Lunch is coming up next. No. We have a great star for our last session before lunch. I do want to say one thing about lunch before Sir Adrian Hill speaks. We have 3 questions which we're going to put on all the tables. So the plan is, once you leave this room, you'll go out to the buffet, fill your plates up, and bring them back to the room and eat your lunch, meet people, introduce yourself to your table mates if you haven't yet, and then try to work through those questions. We're interested in having a discussion that's transformative, enlightening, and is interesting to everyone in the room. And we'd like you to put on the back side of those questions, which will be on the table, write some of the findings that came out of your lunch gathering. So we're engaging everyone in the room in this activity. And then lastly, we have Sir Adrian Hill here. He spiked our registrations, so I think that there's a lot of people waiting for him to talk, and I don't want to spend a lot of time introducing him. His quote is, "There's never been a better time or opportunity than now to eliminate malaria." Adrian Hill is the director and founder of the Jenner Institute and the Meidel Professor of Vaccinology at Oxford University. The Jenner Institute is named after Edward Jenner, the smallpox inventor, and we have not seen any smallpox cases on Earth since 1987. It's the first disease that's been eliminated through vaccine in humans. So we've been talking a little bit about polio, and Dr. Sabin and Dr. Salk, if they could have been at a session like this in 1955, is sort of like equivalent to having Adrian Hill in the room talking to us about malaria. I think that the R21 vaccine is fantastic, but he's going to talk about some of the new vaccines that are coming out so that we can do even better. Good, better, best, never let it rest until your good is better and your better is your best. Here's Dr. Hill.
Wonderful. Well, thank you for that flattering introduction. I'll try and live up to the eulogy. It's great to be here. It really is, to see the buzz in the room, the excitement, the energy that Rotary brings to malaria control, and hopefully, before too long, elimination and eradication. So, as you've heard, I'm at the Jenner Institute in Oxford, called after Edward, as you've just heard. And I'm going to be telling you a little bit about the role that vaccines will play, I believe, not alone, but in combination with other tools that you've been hearing about, and try and make some guesstimate of what time frame we're looking at. And in the interest of full disclosure, before you hear this, I am an optimist. And I don't think anybody doing malaria vaccines, certainly 25 years ago, would have been considered anything else. I have been working in malaria vaccines since 1993, done about 25 different vaccines, and the one I'm going to talk about that actually worked well is a fairly recent addition to the activity, and delighted to be able to tell you a bit about it. So you know the problem, you've heard all about this, 500,000 deaths a year. Being an optimist, I look at that flat curve and I say since 1990, last 30 years, it's still 500,000 deaths. The optimist's view is there are twice as many kids in Africa today as there were in 1990. Whether that's good or bad, that shows you in a positive spin, the malaria death rate has halved. But we've got a heck of a lot more to do. Mainly of young children, 90% of deaths in Africa, 500 million or so spent each year, quotes, controlling malaria, And vaccines rolled out for the first time, and remarkably, like buses, after having waited a century, I'll show you a slide in a minute, to come along within a year. There's the oldest report I know of, in French, it's quite short, of guys trying to make a malaria vaccine. Interestingly, they were in Africa. Okay, they're French, or were French, they were in Algeria, but they were doing stuff that's not silly. In fact, using sporozoite-based vaccines, you heard a little bit about those, I won't be talking about them, but they were getting some partial efficacy using an approach that's still in development. Now, since then, at least 100, I've tried to count, at least 150 malaria vaccines have gone into arms, human arms, in clinical trials, in approved activities. So that's kind of the bad news. In a century and a bit, we've got just 2 vaccines. So here are our excuses as vaccine developers. It is hard. Why is it hard? Well, there are 5,000 genes in the malaria parasite, in Plasmodium falciparum. In COVID, there are a dozen. There are 4 stages to the life cycle. There are substantial differences in the antigens expressed at every stage of the life cycle. That means if you make a great blood stage vaccine, for example, it's not going to work against the other stages. Harsh, but true. The parasite has involved incredibly ingenious ways of switching different genes on and off. So if you make an immune response to one at the blood stage and you wait, then it's disappeared and a different one has been expressed. Absolutely ingenious. Now, it's taken the parasite, I think, a little time to do that. We estimate that malaria parasites have been around for the best part of 30 million years. How on earth could we know that? We weren't here, you weren't here, even the old people weren't here, right? The answer is you look at the variability in the blood stage parasite of the sequences of key antigens, and using a molecular clock, DNA has a clock, we can calculate back roughly how old the malaria parasite was when it started diversifying. And the answer, astonishingly, is 30 million years. So we've co-evolved with malaria. Arguably malaria, well, definitely malaria existed before there were human beings, but there would have been a hominoid ancestor. And that's probably why it's come up with such ingenious solutions to deal with our immune system and carry on transmitting when we think we've developed immunity. So one of the many things that I say or could say when people ask me, why is it so hard to make a malaria vaccine? I say, well, have you seen any other parasite vaccines? There aren't any licensed. Probably the main reason is that the antibody response you need to make a malaria vaccine is many-fold higher, not 25 micrograms per mil, which protects you against COVID. There were lots of those vaccines that got to 25 micrograms per mil. We need about 150 micrograms. And we found that out the hard way by making loads of vaccines that didn't get to that level. And that's just focused on one malaria antigen. So technologically, this is much more difficult than, I would argue, all the vaccines on the market, including COVID, which was relatively straightforward and made quickly. So, what are the 3 key stages of the life cycle? I'm leaving out the 4th, which is at the liver stage, which I worked on for years, and you need T cells to protect against. That is really difficult. If you think antibody-inducing vaccines are, this is much more difficult. So we have 2 vaccines against the sporozoite stage, the parasite injected by the mosquito. Not sure if I can point on this. Oops. No, that takes me backwards. Yeah, sorry, I need this. So, sporozoites injected by mosquitoes go very quickly to the liver after being in the skin for some hours, hang out in the liver, of course, for at least a week or for a week, and then infect the red cells, and then you begin to become unwell. And if your immune system can't deal with that, you die of malaria. If you take antimalarial drugs, that should help. And then there are different antigens here with the red cell stage to what you had at the sporozoite stage. And then the 3rd stages are these sexual parasites, literally males and females, and they mate in the mosquito, of course, and the life cycle transmits. So what the world has at the moment is R21 and RTS S, which I'll come to in a moment, which is the first licensed malaria vaccine, just. Blood stage vaccines like RH5, and this antigen is remarkably conserved. I told you earlier there's great genetic diversity in blood stage parasites. This is an exception. I'll come to that. And then different antigens inside the mosquito. And what you're trying to do here is to vaccinate people so that antibodies are taken up by the mosquito bite as well as the parasite, and they can't mate inside the mosquito midgut, and you cannot continue transmission. So of those approaches, sporozoite stage is in the lead with these two vaccines, relatively similar, but actually, I'll argue, importantly different. So the first malaria vaccine was RTS S, made in 1984 by Joe Cohen in a lab in Belgium, in Rixensart, working for a company then called SmithKline Beecham. They were collaborating with the US military fairly extensively, who funded a lot of the activity. And the trick that Joe came up with was to use the hepatitis B vaccine that most of you have had. It was made by the same company in Belgium. And the advantage of doing that is that if you put a bit of malaria, and this is a key malaria protein, circumsporozoite protein, on hepatitis B, you cover the surface of a nanoparticle. What's a nanoparticle? It's a tiny sphere, 20 nanometers in diameter. And if you stick malaria antigens onto that, we and lots of people have shown, you get stronger immune responses. And remember, problem A in making a malaria vaccine is getting high enough antibody levels, okay? Why did he put in all the excess of hepatitis B? There are 4 molecules of hep B to 1 of the fusion protein. Well, in 1984, expressing that in yeast, it just wasn't technically possible. Move forward to 2011, and a graduate student in my lab, Kath Collins, was able to order from a commercial supplier a different yeast where you can get this to assemble into a nanoparticle. So let me show you what that looks like. Relatively similar, except in the 1984 version, you've got little islands of malaria in this color on the blue hepatitis B surface. And in 2011, and since then, we've been able to make a totally coated protein. So there's malaria on the outside, hep B on the inside. If you ask, does it work as a hepatitis B vaccine? The answer is yes, but there's an even cheaper one of those around, and you've probably had it. But the two vaccines are now different. This is a real improvement. You can get away with 5 micrograms because it's mainly malaria. Here it was 25 micrograms. That matters for a low-cost vaccine, and this is much lower, less than half the price, about a third at the moment. This has higher efficacy. Very important, I'll show you that in a minute. It lasts longer, which you tend to get with higher antibody responses. We know that the antibody levels induced by R21 are 2 to 3-fold higher than with RTS. That's really recent data, not published yet. And it's easier to manufacture, so there's 20-fold more supply available at the moment. The other thing you need to make a vaccine work, particularly a protein vaccine, is a thing called an adjuvant. Latin adjuvare, to help, increases the antibody responses, and the two different adjuvants used for the two vaccines are related, and they're about, in my view at least, very similar in potency. Probably the most important thing we did in this whole vaccine development campaign over the last 30 years was hook up with these guys. The Serum Institute of India is the world's largest vaccine manufacturer. It's the major supplier by far of vaccines to Africa, and they don't do small scale. That's what the photo is telling you here. These are many hundreds of liter bioreactors in a single factory in Pune in India, and that allows them, simply because larger scale means lower cost, allows them to sell vaccines very inexpensively. So we made a vaccine in Oxford, the R21, did phase 1, it looked safe in the UK, infected people deliberately with malaria who had been vaccinated. The efficacy looked around about 75% in small numbers, but moved on quickly to sites in Africa, Kenya initially, and then a key trial led by Haladu Tinto in Natnoro in Burkina Faso, VAC 76, and it was quite small. only 150 children, a control group, group 3, 2 different doses of the adjuvant to see which was better. The higher dose turned out to be better. And to our delight, we got the highest efficacy of any malaria vaccine over the last 100 years at first shot. Group 2, with the higher dose of adjuvant, 77% efficacy in the target age group of young African children. The age, as you probably know, you're most likely to die of malaria is 1. After a year, you've developed a bit of immunity. After 20 years, you've developed a lot of immunity, and you probably won't die from malaria unless you're getting pregnant or you're really unlucky. So here's the impressive curves, and we were so encouraged by that we went straight away to a phase 3 trial, and that's just showing you that the 77% is higher than the equivalent result for RTS S, which has held up very well. And in fact, if we followed it over 4 years, as we did the same children, yes, the efficacy dropped by about 12% to 64%, but that's still the best result with any malaria vaccine. The phase 3 trial was led by 5 really outstanding African scientists in East and West Africa, in Kilifi and Bagamoyo, as well as in Buguni in Mali and in Burkina Faso. Our clinical lead was Maureen Datu from our institute in Oxford. And 4,800 children were now recruited, because you need a lot of safety data if a regulator is going to approve your vaccine. So we moved ahead, and to our delight, we were able to replicate that result in phase 2 in a large phase 3 trial, 79 or 75% efficacy, depending on the local ecology of malaria. And with that data, having followed up for several years, we got approval from the WHO, and actually, interestingly, from Ghanaian and Nigerian regulators about 6 or 7 months before WHO approved the vaccine. But in terms of elimination, this was the most intriguing result. We weren't setting out to make a vaccine that would help with elimination. Antisporozoite vaccines can help, probably, but they're not the sole tool. But interestingly, just at 6 months or at 12 months, most of the children had a more, well, on average, the reduction in having parasites in your blood as a vaccinee. In other words, vaccine efficacy against asymptomatic malaria. These children were not unwell. We cut down the rate of carriage of parasites in the blood. And as you know, in Africa, lots and lots of people carry parasites and are totally well. So would this help with elimination? And we started thinking more about that. Just to summarize where things are around about now, delightfully, most African countries with very high malaria burdens are receiving vaccines. These are the ones where R21 is distributed. These are the RTS S ones. But the scale of distribution this year is very different. 100 million doses for R21 and 6 million doses for RTS S. It's not that GSK don't want to produce more. They're really limited by access to adjuvant, and that adjuvant that they own is used in shingles vaccines and other vaccines you may have had. So how to progress from where we are? What we would really like is a 3-stage malaria vaccine. I'll explain why. Rh5 is a leading blood stage antigen that my former graduate student, now professor in Oxford, Simon Draper in the biochemistry department, has identified and has now developed to phase 2 trials and is looking at phase 3 trials. And he has seen very interesting efficacy in phase 2, about 55% against clinical malaria, and even higher efficacy against more severe malaria. And if you think about that, that makes sense. If you're protecting at the blood stage, stopped replication in the blood, you'll get better efficacy. Lots of work also on transmission blocking vaccines, several groups at the NIH, Patrick Duffy particularly, and also Shumi Bijouage in Oxford. 3 candidates fighting to get to the finish line. I don't think you need a winner. I think if you had a mixture of all 3 of those, you'd probably get even better transmission blocking. We'll find out, hopefully, in the next couple of years. And elimination trials are actually happening as we speak with vaccines. In Bangladesh, where transmission is relatively low, not a silly starting point, but also in the Gambia, where results were released in the last few days by Humberto D'Alessandra's group, and also in Burkina Faso in the same trial. That trial is called SERVAL, and because time is short, I'll just say that vaccine efficacy, particularly in the Gambia, has been very high in adults, in a very large number of adults, including some women who will have become pregnant, as well as in children in the Gambia. So that's pretty encouraging. But this is where we want to be. Well, we are now, but we want a blood stage and a transmission blocking vaccine licensed. So we have R21, we're pretty happy with that. RH5, there's the efficacy in Burkina Faso. That needs to move into phase 3 with or without R21. And then these complicated names are 3 different transmission blocking vaccines that all show promise based on some in vitro antibody assays. So where are we going? Well, where I was going a couple of weeks ago was Bioko Island in Equatorial Guinea, one of the wealthier countries in Africa. It has oil, it has gas, and it has pretty good epidemiology since 2004, showing that they have brought down parasite rates very substantially, 70 to 80%, but not down to zero. Guillermo Garcia with MCD, the American Foundation here, have been working with traditional tools to control malaria. They're very excited about vaccines. They brought us to see the Minister of Health, who was also excited about vaccines, and we're thinking whether we can do this in a feasible way with vaccines and possibly one more tool that I'll come to in a moment. Similarly, Roly Gosling, London School of Hygiene and Tropical Medicine, is starting a trial not of 1 million people in Zanzibar, but of the young males who party at night or just stay up at night and are the most susceptible population. And they're going to use antimalarials with a vaccine. So things are beginning, but we're not there yet. Just to remind you, the steps are to optimize a low-cost, high-availability, 3-stage vaccine. So you could say we're only a third of the way. But really excitingly, you heard this was actually hiding on one of the slides that we saw in the last talk, MMV371, one of these new long-acting injectables, drugs that don't last a day, so you have to take drugs on day 1, 2, and 3, but last months. And MMV are looking at, oddly, a trial in Nottingham, how long this drug lasts. And it's not weeks, it's months. It's at least 3 months from the data I've seen on one of Christine Donini's slides. It could even be 6 months of efficacy. That would be a complete game changer for malaria control. Just as you may have heard of the long-acting monoclonal antibodies that are, sorry, not monoclonal antibodies, long-acting drugs for HIV, lenacapavir is the lead, that lasts for 6 months. And 2 shots of an injection of that drug keeps your viral load pretty undetectable. Can we do the same in malaria? Well, it's looking very good. My view is that we will need one of these drugs in combination with vaccines to finally get rid of malaria. So you should ask MMV how long we are from having a licensed MMV 371. I hope it'll be sooner rather than later, and no pressure there. So technically, I think malaria elimination looks feasible because of these new tools that have come along just in the last few years. But just to remind you of some sums that are not in dispute, but we tend to forget. And of course, the problem is we're trying to provide tools that are new and therefore might even be expensive, but shouldn't be expensive, for vast numbers of people in the poorest continent in the world. But the sums add up and you win. So let's just review this. This is Malaria No More. Lots of people have made the same point. Jeffrey Sachs, 25 years ago, that the cost of the control of malaria, which we know is about 5 billion at the moment, could be cut very substantially if you could eradicate malaria, because you'd stop and save your 5 billion. But much more importantly, all the estimates and all the health economists believe that the GDP of Africa will and should improve, and not by a trivial sum. The estimate here is 16 billion a year, right? And you never have to pay for vaccines for malaria again if you've eradicated it. So vaccines and other tools that will eliminate malaria are a bargain. And yes, I know the problem is that this is money afterwards, not money beforehand. And we are living in rather strange times in terms of public health funding, to say the least. But this is a win-win. And remember, it was done before. You all know about Gavi. 11 vaccines is what the children in Africa get in most countries at the moment. That's because international organizations came together and created Gavi as a fund to encourage manufacturers like the guys in India who can make very, very cheap vaccines to make them, sell them in Africa at really low prices, and disease rates tumbled. Mortality halved. We should be very proud that vaccines halved mortality over the last 25 years in Africa after Gordon Brown and lots of other people got together to make sure the world had cheap vaccines. We need something like that for malaria. And remember, in this case, you definitely got your money back if you increase GDP in Africa. So in summary, my thanks are to not just the 5 amazing principal investigators in the phase 3 trial, but to them and their huge teams, to my colleagues at the Jenner Institute in Oxford. This is a small number of the lead investigators there, and particularly to the Serum Institute of India. Thank you very much for your attention.